Thank you for the introduction. Hello, everyone. Thank you for being here for my presentation entitled Nontraditional Decontamination Methods for Salmonella Reduction in Dried Fruits and Cereals. As mentioned, my name is Kayla Murray, and I'm a PhD candidate at the University of Guelph in Ontario, Canada, and I hope you're enjoying my presentation today. The outline of the presentation will be as follows. First, I will be discussing cereal and dried fruits and the pathogen of concern and the outbreaks associated with it, the sources of the pathogen within low-moisture food items, and the non-traditional decontamination methods used. And then I will open up the floor to any questions you may have. In regards to cereals, we have two main basic categories: whole grains themselves, such as rice and barley, or their flour-based derivatives, such as bread, pasta, sweets, and of course, breakfast cereals, and flour itself. In terms of dried fruit, there's a great variety of them, which include, most popularly, raisins, followed by dates, prunes, figs, and apricots. In terms of water activity and microbial growth, low-moisture foods are classified as having a water activity of 0.6 or less, as you can see on the left here. This level of water activity is not sustainable for microbial growth or spore germination, as shown on the right, but this does not necessarily mean low-moisture foods are microbiologically safe, however. This just means that whatever they became contaminated with along the harvesting, processing, transporting will not continue to grow in the final low-moisture food product. However, if the contaminated low-moisture food product is being added to a high-moisture food product, there is quite a significant microbiological risk, which I'll discuss a little bit later on. These are some of the products we know and love: breakfast cereals, cereal bars, milled flours, and dried fruits. And these are also the products that have been associated with recalls and outbreaks in North America due to salmonella contamination. When analyzing the burden of illness related to low-moisture food outbreaks, salmonella is dominating the field. Salmonella was responsible for nearly 45% of the outbreaks, leading to nearly 900 hospitalizations and unfortunately leading to 14 deaths. Bacillus cereus, which was the next highest pathogen associated with low-moisture outbreaks, being at 25.7% of the outbreaks, but fortunately leading to very few hospitalizations and 0 deaths. Looking at the number of outbreaks of low-moisture foods, we can see that grains and cereals make up the greatest number of outbreaks, and that is mainly due to Bacillus cereus, where we're seeing spore germination followed by the thermal shock of the item, which leads to the outgrowth in the cooled product. Salmonella, however, is also of concern, with a relatively low infectious dose of around 100 to 1,000 cells, and in some serotypes we're even seeing as low as 10 cells, research shows. Therefore, it is vital for ready-to-eat low-moisture foods to be completely free of it for our consumers to be safe. As we likely know, Salmonella is a gram-negative, non-sporulating rod. It is a facultative anaerobe with a growth range temperature of 5 to 46 degrees Celsius, with a minimum pH for growth of 4.5 and a minimum water activity of 0.93. Salmonella is shed through fecal matter, which can cause salmonellosis, which has gastrointestinal symptoms including fever, diarrhea, nausea, cramping. Usually these symptoms occur within 8 to 72 hours and last a few days. For healthy individuals, that is. Unfortunately, for immunocompromised individuals, the infection can be much more serious. As mentioned, Salmonella sheds through animal and human fecal matter, so unfortunately it can become a contaminant of many sources. Most notably, we are seeing contamination due to poor worker hygiene, which can be an issue at nearly every stage of production, from cultivation, harvesting, storage, transporting, as well as processing. Irrigation water used in cultivation can also be a source of Salmonella, as well as the equipment used throughout the processes. The outgrowth of the pathogen within the dry processing environment can be attributed to the presence of water. Within the product itself, the outgrowth depends on many factors in the process, most notably the amount of time it takes for the high moisture food item to become in a less suitable environment and become dry as a low moisture food item. The drying process itself also plays a factor in regards to the thermodynamics of the process. And lastly, maybe most importantly, is the maintenance of the eventual low moisture products after the thermal shock process, in regards to maintaining that low moisture product, as well as making sure it doesn't come into contamination after the thermal kill step. Although we know Salmonella cannot grow in the final low moisture environment of these foods, it is able to survive and adapt to these conditions through different mechanisms, which I will speak briefly on. The mechanisms mostly involve the altering the osmolarity of the internal cell compositions in order to be in balance with the external low moisture environment to prevent desiccation. The process of cell osmoregulation are summarized in this figure. For example, the cell can accumulate electrically neutral, low molecular weight osmoprotectants such as glycine, betadine, ecotine, and proline. These solutes are able to limit the water loss of the cell without affecting the cellular functions. Transporters Pro-U and Pro-P are also important in this mechanism, and studies have observed that an upregulation of these genes during the early stages of desiccation. Other genes have also shown to increase during the early signs of desiccation, including the KDP genes. In addition to utilizing osmoprotectants, Salmonella is readily able to enter a viable but non-culturable state in response to stress, further ensuring its long-term survival under the low-moisture food products. As well, it is hypothesized that biofilm formation may play an additional role in survival. So we know Salmonella can survive for long periods of time in a low-moisture environment. But how does the thermal treatments that cereal and dried fruits undergo affect the survival of Salmonella? Research shows that Salmonella is easily destroyed in high moisture food items with the thermal kill cycle. But once the drying has occurred, the efficacy of this measure proves very challenging. Even at a moderately low water activity, studies have shown that Salmonella have an increase to heat tolerance to the extent of which varies between food products and depends on other intrinsic and extrinsic properties such as pH and oxygen availability. Moving on to intervention methods, I'm going to be discussing three main categories of decontamination technologies. First, chemical, followed by thermal and non-thermal. So we know that traditional thermal pasteurization and disinfection methods are not suitable for low moisture foods due to the increase in heat tolerance that comes along with being in the low moisture environment. Hence the need to implement non-traditional methods. The methods which would be ideal for low moisture foods keep the food items in the processing environment dry, which is super important, or else there's a risk of recontamination after the intervention methods. The treatments would yield no to little or acceptable levels of residues. They would be ideally cost-effective and have been validated to show a 5-log reduction. And most importantly, they would be safe for the personnel involved using them. First, let's discuss chemicals, which can be used in the gaseous phase, which satisfies the first requirement of being a dry intervention. Ethylene oxide is used in some countries as a pesticide and sterilizing agent. The chemical works best under dry conditions, destroying most viruses, bacteria, spores included, and fungi, making it an ideal dry cleaning sanitizer within low moisture food processing environments. On foodstuff itself, ethylene oxide is only permitted in Canada to be used on spices. The levels of which is permitted must align with good manufacturing practices. However, the byproduct residue, ethylene chlorohydrin, may not exceed 1500 ppm. The treatment also must be followed by an aeration period in order to ensure all the gas is dissipated from the product. The downfalls of this sanitization method, unfortunately, include it being carcinogenic and having quite explosive properties. Propylene oxide, which much like ethylene oxide, is a highly volatile chemical which has been used as an insecticide and antimicrobial fumigant in dried fruits, nuts, and spices in the US for over 50 years, with a maximum residue level of 300 ppm. In order to import into Canada, items must have a general maximum residue level of 0.1 ppm or less. It is classified as being possibly carcinogenic to humans by the United States Environmental Protection Agency with an occupational exposure limit of 2 ppm. Ozone has been used in different industries for a long time, especially in municipal water treatments, as well as bottled water production. I'll speak briefly on it before moving on to thermal and non-thermal interventions. More recently, ozone has become popular for the direct application and indirect application on food items. It is a very broad spectrum biocide and often used as an alternative to chlorine. Ozone is usually formed by corona discharge, which essentially mimics the way lightning is produced in nature. This process is where dry oxygen is passed through an electrical field, causing the oxygen molecules to first split and then rejoin together as a triatomic molecule. The second most common method used in ozone generation is using ultraviolet light at 185 nanometers to produce the same effect, however, at a much lower concentration than with corona discharge. The ozone produced by these methods is able to rupture cell walls of organisms, proving to be very effective. The levels permitted to use on food items must be in accordance to good manufacturing practices, and unlike other chemicals I've spoken about, ozone has a great advantage of not leaving residue byproducts. However, workers must be very cautious when working with ozone to prevent damage to the lungs. There are many alternatives to gas-phase chemicals we discussed, and those include irradiation, radiofrequency, gas plasma, dry steam, and advanced oxidative process, which Nita talked about, and LED UV. I'll be going through some different examples and how they can be used in low moisture product environments. Irradiation has been commercialized since the late 1950s, but was only used for sterilization of medical devices and disposables until 1992, when irradiated foods became commercialized in the United States. At least one food product in over 35 countries have been approved for irradiation. Most commonly observed is for the use of spices, followed by fruits and vegetables. Depending on the dose of radiation applied, foods may be pasteurized to reduce or eliminate pathogens, or for some particular uses, food-made items may be sterilized to eliminate all microorganisms, with the exception of some viruses. Low to medium doses of 1 to 10 kilograms are most commonly used, which kill insects and larvae in wheat and wheat flour and destroy pathogenic bacteria and parasites. Low to medium doses also inhibit sprouting of potatoes and other foods and slow the ripening and spoilage of fruit. Higher doses, between 10 and 50 kilograms, sterilize foods for a variety of uses, such as for immunocompromised hospital patients or astronauts. There has been much public concern over irradiated foods, unfortunately, and the byproducts formed, likely in part to this symbol that has to accompany food packaging, implying that the food needs to come with some sort of warning label. In reality, studies have shown there to be no unique radiolytic products of toxicological significance in irradiated foods. The byproducts found are typical of any cooked food. The real drawback of irradiated food is, like with cooking, some vitamins are sensitive to the treatment, as well as the method is not known for being exactly cost effective. Radiofrequency utilizes wavelengths of 3 gigahertz to 300 megahertz to penetrate and rapidly heat solid and semi-solid food items within their packaging. This is due to a direct interaction between food items hermetically sealed in packages with electromagnetic waves. The wavelengths used are much longer than the wavelengths created via microwaving and hence penetrate much deeper into the food product. It is very interesting technology because the heat is generated directly within the product at a molecular level and therefore doesn't require heat transference in contrast to traditional heating. This has the benefit of avoiding alterations into the sensory and textile properties of the food due to its very rapid nature. Ultraviolet light has been used for many years in different industries to inactivate microorganisms, including for water purification and pharmacological purposes. It has a relatively low penetration rate, so it is ideal for flat surfaces, air, and transparent liquids. On the electromagnetic spectrum, we have UVA, which is from 320 to 400 nanometers, UVB, which is from 280 to 320 nanometers, and UVC, which is from 200 to 280 nanometers. And there's UVB, which is from 100 to 200 nanometers. Most effective from a bacteriological standpoint is certainly UVC, especially from wavelengths of 250 to 270 nanometers. UV light can inactivate bacteria, viruses, cysts, and molds, as well as yeast. Photon absorption by microbial DNA at wavelengths from 240 to 280 nanometers induces DNA damage and sublethal damage to DNA repair systems. Traditional ways of producing UVC is to utilize high voltage arc discharge mercury or amylogen lamps, which generate photons solely at 252.7 nanometers. However, there are several drawbacks to these traditional methods, mainly that they are not food plant or food preparation area friendly. The equipment can be expensive to build and difficult to maintain and clean. And they do not, unfortunately, operate efficiently under cold room temperatures. And of course, mercury exposure is recognized as a danger to human health. This is why UVC light-emitting diodes, or LEDs, have been explored as of late for low-moisture foods. LEDs are robust and small, they require no warm-up time, and contain no toxic elements. And additionally, they are very low cost and energy efficient, with efficiencies up to 80% versus the typical 35%. And they can operate efficiently at low temperatures. They have a much longer lifetime than traditional method of generation, lasting up to 100,000 hours versus the traditional 1,800. But again, we're seeing the drawback with low penetration within the food items. In conclusion, to wrap up the presentation, it is clear that low moisture food products represent a food safety hazard risk, especially when their low moisture foods are added to high moisture food products during the processing. As mentioned, there are many points in the chain where the products can become contaminated with salmonella, and this is especially dangerous due to the fact that salmonella can withstand dry environments with an added heat tolerance. Fortunately, there are many decontamination methods being researched and adapted to low moisture food items themselves and within the processing environment. With the recent outbreaks and recalls within North America, new regulations are very likely to take into account the methods of these decontamination methods. Thank you so much for your time. I hope you found the presentation informative, and I'll now open up the floor to any questions you may have.